All right, hello. Hi, can everybody hear me? We'll go ahead and get started with today's lecture. My name is Michelle Giarmarco. I'm a fifth-year grad student here in the Biochem department, and for the last 4 years, I've had the pleasure of working in Dr. Hurley's lab, doing some really cool research, imaging the retina, and trying to answer questions about its metabolism. So you can ask me questions about that after class if you want. Today, I'm here to give a guest lecture about glycogen, awesome molecule, as you will see. This is what we're going to do today. I'm going to tell you what glycogen is, I'm going to tell you how we break it down, how we make it, and then what happens if you can't break it down or make it. So glycogen, what is it? You guys have been hearing a lot lately about glucose and how it's an important fuel for cells. And you also heard lately that starch is a polymer of glucose that plants use to store glucose. And like starch, glycogen is also a polymer of glucose. It has a very similar structure, as you'll see, and it is used by plants or animals and fungi. So I like to think of glycogen as like the starch for animals and fungi. This is what it looks like. It's very beautiful. It has a protein at its core and is surrounded by all of these branches of glucose chains. And it kind of looks like this creepy sea star. It has a very organized, almost fractal-like pattern with all of these intricate branches and lots of things going on. But the cell doesn't just do this to make a beautiful thing. There's actually a use for having this polymer, have this structure. So can you guys think of what would be an advantage of polymerizing glucose like this? Yeah. Yeah, it's an efficient use of space. I like that. It concentrates the glucose, right? It takes, if you have a drop of water, you imagine, that has 0.01 micromoles of glycogen in it. This is like unimaginably small, actually, but if you were to dissociate all of that glycogen into its free glucose, you would have 400 millimoles, which that's like a 10,000 factor of concentration, which is pretty huge. And the benefit of this is that glycogen is insoluble. The problem with storing all of this glucose in a cell is that it's soluble in water. So wherever glucose goes, water goes, and you can kind of think back to your early biology class where you had the cell in a beaker of water and it was hypotonic and the cell exploded. That happened because the osmotic pressure goes crazy, and the same can happen with having too much free glucose in the cell. So glycogen lets the cell store a lot of glucose without affecting the osmotic pressure very dramatically. And in other words, it keeps the cell from exploding. Just for some reference, the concentration of glucose in your blood is around 5 millimolar. So this is like a really efficient way to concentrate glucose. We can also see glucose on an electron micrograph. So this is, yes, electron micrograph image of a liver cell from an animal that is well fed. And all of these peppery spots that you see surrounding it are glycogen granules. These are mini compartments, so it's not quite an organelle, but it's like an aggregation of 20 to 40 glycogen molecules and all of the enzymes that are associated with it to break it down and make it. So they're pretty large, on the size, roughly around the size of a ribosome. And yeah, they're a very efficient packet to store and release lots of energy. And also what's really cool is if you take the same image and you look at an animal that has been fasted, all of these glycogen granules are gone. So this pool of glycogen is very dynamic and very much alive, kind of depending on when your last meal was. You also may be thinking to yourself, like, it's the holidays, fat has a lot of calories, our bodies are really good at storing it. Why does the cell go to the trouble to make this other molecule, and why doesn't it just store this all as fat? And you guys know that fat is insoluble and in water, and this presents challenges for its synthesis and breakdown that you're going to be hearing a lot more about next quarter, but suffice to say, the breakdown of fat is slow. So in this way, fat kind of contributes to longer-term energy homeostasis if there's prolonged fasting or starvation going on. This is in contrast to glycogen, which, because it's packaged so efficiently in these granules, can be broken down and made very rapidly. And so because of this, the body can use glycogen to meet kind of acute energy needs. So if the blood sugar is too low, or muscles need glucose to contract, to make ATP to support their contraction, glycogen is going to be there almost like a battery of a readily available, quickly available fuel source they can tap into, which is in contrast to fat, which is more like coal. It contains a lot of energy for sure, but it's going to take a long time to break it down and actually get all of that energy out into a usable form. So where is glycogen? We already mentioned liver cells, and it's actually found in many different tissues in your body, including the brain and even the retina. Most of it in humans is stored in liver and skeletal muscle, and you're going to see that it has very different roles in these two tissues. It comprises about 10% of the wet weight of liver, which is a lot, and only about 1% to 2% of the wet weight of skeletal muscle. But if you think about how much muscle you have relative to liver in your body, you will realize that about 75% of your glycogen is actually stored in the muscle. I mentioned they have different roles. The liver is very altruistic with its glycogen. It breaks the glycogen down all the way into glucose, which it then exports into the blood so that other tissues of the body can have it. So the liver is very charitable in this way. It has a exportable glucose reservoir in the form of glycogen. And depending on your activity level, this store of glycogen can last for 12 to 24 hours. This is in contrast to skeletal muscle, which is a lot more selfish with its glycogen. It doesn't even break it down all the way into regular glucose. It stops at glucose 6-phosphate, which is a famous intermediate that you guys have heard a lot about lately. It can enter glycolysis, it can enter the pentose phosphate pathway, and sure enough, muscle uses this to go straight into glycolysis to generate energy. So muscle uses its glycogen as a usable energy reservoir for itself. And this supply of glycogen can be depleted in about an hour of rigorous exercise, but it's very, very useful when your bus is pulling away from the curb and you have to run to catch it. So the structure of glycogen, we'll dig into this a lot more. I showed you already this protein at the core called glycogenin, and then it's surrounded by all of these branches. So if we zoom into these branches, it looks like something that maybe you guys recognize, a carbohydrate. It has an anomeric carbon down here, but in glycogen, none of the business is happening at the anomeric carbon. All of the activity is happening at the non-reducing ends. This is where glycogen is polymerized and depolymerized. Because of that, there are two carbons available that can participate in reactions depending on which enzyme is around. So glucose units can be added at either the 4 or the 6 position of that most terminal non-reducing end glucose. And because of that, there's two different bonds that can result. We can form an alpha-1,4 linkage between carbon 1 and carbon 4 of two different glucoses, and that forms a linear chain. And then we can also alternately form alpha-1,6 linkages between carbon 1, carbon 6, and this forms the branches that we were talking about. And we can kind of compare this to what's going on with starch. Starch is a mixture of alpha amylose, which is not branched, so it has all alpha-1,4 linkages. And then it also has amylopectin, which is branched, but only about a third or half as much as glycogen is. So why is glycogen so highly branched? What is the benefit of having all of these branches? Does anybody have any stabs? Yeah. Yeah, you can get more glucose more quickly and you have lots of branches to pull it from. Exactly. That is totally right. There are 2,000 non-reducing ends on a single glycogen molecule at any given time, so you can essentially have 2,000 reactions going on if you have enough enzyme. So that means that glucose can be really rapidly released, and the benefit that this confers to us animals is that we can sustain muscle contraction. Plants have an inferior form of glucose storage, starch, so therefore they can't actually walk, only in cartoons. So one reason why plants can't walk. That was the overview. Now we'll talk about breaking down glycogen, otherwise known as glycogenolysis. It's a very logical name. What would you call the enzyme that breaks down glycogen? We can't really talk about this pathway, though, without first giving a shout out to this amazing husband and wife scientist team, Carl and Gertie Corey. They worked in a dark age of science when things were kind of scary and carcinogenic, and it was very much a new frontier. And using the state-of-the-art that they had available to them between 1925 and 1950, they were able to elucidate most of this pathway that is still published in your textbook today, which I think is amazing. They had, crude 1930s methods, so they just minced up some frog muscle and were like, what metabolites are in here? They found a lot of this intermediate, glucose 1-phosphate, and they did what was the fashion of the time for scientists. If you discover a cool new compound, you name it after yourself. So they named it the Corey ester. And this intermediate has become so important and so pivotal in this pathway that it's been featured on a postage stamp. The Coreys went on to win a Nobel Prize in 1947 for their work. They've trained a bunch of other Nobel laureates, and they've helped to identify and characterize several diseases that are related to this pathway. So not to make the rest of us feel bad or lazy, the Coreys are awesome. This pathway is pretty simple in terms of how it works. Single units of glucose 1-phosphate are just chipped off of glycogen one by one. They are then converted to glucose 6-phosphate. That intermediate can then enter a number of pathways depending on what tissue it's in. So if it's in muscle, it can go into glycolysis, like I said, to generate fuel. If it's in liver, it will run into this enzyme glucose 6-phosphatase. And this enzyme is the key that gives the liver its altruistic glucose exporting powers. This enzyme will dephosphorylate the glucose 6-phosphate into just plain glucose, which then gets exported. Muscle lacks this enzyme, so therefore muscle can't be so charitable with its glycogen. So there are three enzymes, luckily for you guys, only 3 that are involved with this reaction, and the pathway is elucidated over here in a nice cartoon form. First, glycogen phosphorylase cleaves those alpha-1,4 linkages, so those are the red hexagons up here in this diagram, again from the non-reducing ends, and it'll just kind of chomp along all of one of those alpha-1,4 linkages until it gets to 4 units away from a branch, at which point it's almost like its arms are too long to reach in or too short to reach in and catalyze the reaction. And so another enzyme picks up, debranching enzyme, which has 2 jobs. It first transfers the 3 residues in blue onto the end of the chain to kind of linearize it, get rid of most of that branch, and then it has a glucosidase that cleaves that last alpha-1,6 linkage to yield glucose. And then we still have to deal with this glucose 1-phosphate that we made up at the top, and so there's an enzyme called phosphoglucomutase that converts that to glucose 6-phosphate, so it can be used. And we'll be talking about all of these enzymes more in depth, but I kind of want you guys to appreciate that these pathways are they're often presented in this kind of a linear way, but all of these reactions are happening simultaneously, and like the singers in Destiny's Child, all of them have to be on their note in order to achieve harmony in this pathway. So, and maybe one of them gets a little bit more attention and is slightly more talented. So glycogen phosphorylase, I think this one is Beyonce. It's really, really cool. It breaks down glycogen, again, at the alpha-1,4 linkages to yield that Corey ester glucose 1-phosphate. And what's special about this reaction, maybe you notice, is that it's using phosphate to run this reaction. So far, hydrolysis has been the traditional method of carbohydrate breakdown that you guys have been learning about. This is using phosphate. It's a process called phosphorylasis. And yeah, why phosphorylasis? Why doesn't this enzyme just use hydrolysis like the rest of all the enzymes? You guys have any ideas? All right, well, one reason is that you don't need ATP to do this reaction. You can phosphorylate something without adding ATP, which is pretty clever. This phosphate is very abundant in the cells, so this whole reaction is kind of like a free ticket energetically. And then also, Once glucose is phosphorylated, it can't exit the cell. So I think you guys heard hexokinase kind of has a similar function. Once it phosphorylates the glucose is like trapped in the cell. It's a similar idea here, although this reaction is generating glucose 1-phosphate directly. So essentially, there's just less chance for it to be lost to export or other reactions that might be looking for bare glucose. Glycogen phosphorylase also has some unique relationships with its cofactor, pyridoxal 5-phosphate, or PLP. This is a derivative of vitamin B6, and it's an essential cofactor for this enzyme, which means the enzyme won't work without it. This also means that people that have vitamin B6 deficiency can have problems with glycogen handling and blood sugar, and this is the reason why. Here is glycogen phosphorylase with the PLP in magenta sitting in at the active sites there. And what this diagram is really trying to draw your attention to is this lysine 680 residue. If we look at this, we see that the cofactor is covalently bound to the lysine 680. So down at the bottom is the PLP, at the top is the enzyme glycogen phosphorylase with its lysine 680 showing. And there's a Schiff base that's linking these two together. And why this matters is that not only is it covalently linking the cofactor there so it can never get away, but it also leaves the phosphate of that PLP free to participate in reactions, and that's exactly what it does. So the PLP acts as a prosthetic group, so that phosphate is actually participating in the catalysis of the mechanism of the reaction, which looks like this. The color scheme's just a little bit different now. PLP is blue, the phosphate is red, and then glycogen is black with its non-reducing end glucose showing there. And this is essentially just a series of proton exchanges from PLP to phosphate, from phosphate to glycogen. This makes glycogen, the rest of glycogen chain, a good leaving group, and you're left with this glucose carbocation intermediate here. This exists for a short time because it soon finds the negative phosphate on PLP, forms a covalent bond there, and maybe you can start to recognize that thing on the very end as our Cori ester glucose 1-phosphate. So it's a very well understood mechanism for this reaction. There's also an inhibitor called 1,5-gluconolactone, and does anybody know how this thing might inhibit the enzyme? Well, if you remember from organic chemistry, or maybe you're almost to organic chemistry, this carbonyl carbon here in the inhibitor has a slightly positive charge. So it just mimics that carbocation intermediate. It can bind up to glycogen phosphorylase and muck up the whole enzyme. So that's a kind of a nice mechanism they've got there for this one. Debranching enzyme is not as well understood, although interestingly in mammals, it's one enzyme with two different active sites performing these two very different jobs. So it has a transferase domain that again moves those 3 glucose units that are blue on the top from the branch onto the main chain. And then it has the alpha-1,6 glucosidase domain that cleaves off that last alpha-1,6 linkage, and that yields unphosphorylated glucose. You should note that this glucose is unphosphorylated. It has to be phosphorylated before it can do anything biologically useful with its life, which consumes energy. So just note that. Also, this end now is open for phosphorylasis. So debranching enzyme and glycogen phosphorylase are kind of hanging out together in the same space, and now glycogen phosphorylase can come in here and chop up the alpha-1,4 linkages on that end. The last part of this is the reaction catalyzed by phosphoglucomutase, which has a pretty simple job of just transferring a phosphate from the 1 position to the 6 position of glucose. It does this using a phosphorylated serine residue that actually participates in the reaction, and this is kind of akin to phosphoglycerate mutase you guys learned about in glycolysis, which also used a post-translational modification to catalyze a reaction. Similar thing is happening here. This time it's a serine that's phosphorylated, and that phosphorylated serine in turn phosphorylates the 1 position of glucose. There is a bisphosphate intermediate that exists for a short time, and then the enzyme dephosphorylates the 1 position. Did I say that? It first phosphorylates 6, then dephosphorylates 1. So we get glucose 6-phosphate in the end, which, again, to remind you, if you're in liver, has the magical enzyme glucose 6-phosphatase that dephosphorylates it, makes glucose for export. If you're in muscle, this glucose 6-phosphate just goes straight into glycolysis to make energy. And you maybe also notice that this reaction is reversible. And this kind of provides an interesting point of regulation in that the concentration of glucose 1-phosphate and glucose 6-phosphate kind of determine which way the reaction is going to go. So in the fed state, you have a lot of glucose 6-phosphate right after a big meal, and it's going to drive the reaction toward glucose 1-phosphate. And if you're in the fasted state, the opposite's true. The reaction is going to be driven in the opposite direction to make glucose 6-phosphate to get energy or increase the blood sugar. So that's summary of glycogen breakdown. There's 3 enzymes, 2 products. Glycogen phosphorylase cleaves those alpha-1,4 linear linkages. Debranching enzyme debranches and then cleaves the alpha-1,6 linkage. And phosphoglucomutase converts out glucose 1-phosphate to glucose 6-phosphate. And if you were to take all of the products of these reactions and put them in a bucket, you would have about 92% glucose 1-phosphate and about 8% glucose. Hopefully these numbers should make sense to you based off of all of the ratios of glycogen that we just talked about. And I will reiterate one more time that this glucose, the free glucose, has to be phosphorylated in order to be used. So that is glycogen breakdown. Now we'll talk about glycogen synthesis, otherwise known as glycogenesis. And for a long time, people just thought that making glycogen was the exact opposite of breaking it down. I mean, it seems pretty logical, right? And that was until 1957. This Argentinian scientist had some bright ideas, and he said, hey, you guys, maybe making glycogen is not just the opposite of breakdown. And why did he think this? He wasn't just like some old loon. He actually worked in the lab with the Coreys. He was one of their famous trainees. And he made this important observation that glucose could not be added onto glycogen unless it was first activated with UDP. So he found that out in the Corey lab, and that was kind of their first clue that maybe these pathways aren't just exactly the opposite of each other. And sure enough, he ended up being right. He won a Nobel Prize for his work and went on to work in a number of other pathways and has a pathway named after himself now. So that was a very important observation that kind of led to the rest of this pathway getting figured out. So after the glucose is activated, then it's a little bit more analogous to glycogen breakdown. There's an enzyme that forms alpha-1,4 linkages to build linear chains, and then there's an enzyme that introduces branches. So we're going to talk about all of these more in depth, but there's 3 steps, 3 enzymes. The first step, that activation of glucose with UDP, happens by this UDP glucose pyrophosphorylase enzyme, and this is its reaction. UTP is similar to UDP, except just the nucleotide base is different. Instead of adenosine, it's uracil. And the important part is that this phosphate of the glucose 1-phosphate attacks that alpha phosphorus on UTP. This covalently links the two, so now the glucose is activated. And it also makes pyrophosphate, which seems kind of like a trivial waste product intermediate, but it's actually very important because it provides the energetic driving force for this whole reaction to happen. So there's an enzyme pyrophosphatase that hydrolyzes the pyrophosphate into just plain phosphate, and this is an exergonic reaction, so it releases energy. In contrast to the reaction of UDP glucose pyrophosphorylase, which doesn't really have an energetic incentive to go one way or the other, but because you have the pyrophosphatase reaction consuming all of that pyrophosphate, it shifts the equilibrium toward making UDP glucose in a big way. So that is awesome. Where do we get this glucose 1-phosphate? Initially, it has to come from somewhere, right? Anybody have ideas? Where? I already told you the answer. It's phosphoglucomutase. Remember, it can work in reverse. It doesn't just convert glucose 1-phosphate to glucose 6-phosphate, but if there's high glucose 6-phosphate, it can be used to make glucose 1-phosphate. And then This is kind of an example of a covalent modification being used to activate a metabolite, and you guys have seen some examples of this already several times in this class. Can you think of any other modifications that activate a common metabolite? All right, well, these. Essentially, acetate is just a boring 2-carbon lump until you stick a CoA on it, and now it can enter the TCA cycle, it can get made into fatty acids, it has a life after it has this modification with CoA. And similar, phosphate is just a boring ion until it becomes a part of ATP, and then all of a sudden it's the most important energy currency in the cell. So these modifications, similarly UDP activating glucose to get added onto glycogen, can greatly change the fate of a metabolite. So after we have that the UDP glucose activated, glycogen synthase is the next enzyme that comes in and polymerizes it. This is going to build up alpha-1,4 bonds at those non-reducing ends out on the periphery of the glycogen molecule. And this is fairly straightforward. It results in elongation of an existing glycogen chain. But you might be thinking, you need a glycogen chain first. Like, where does the first glycogen chain even come from to get this whole thing started? And that is where, it's like a chicken and an egg kind of situation. This is where the glycogenin comes in. And this enzyme is crazy. It's a protein, also an enzyme. It's at the center of all glycogen molecules, and it acts as a primer for glycogen synthase. It is an autocatalytic glycosyl transferase, which means that the enzyme is the substrate. So let that break your brain for a minute while you look at this MC Escher drawing of hands drawing themselves. But glycogenin has this special tyrosine residue on it, which it can itself decorate with glucose. So as long as UDP glucose is around, this tyrosine residue, this glycogenin can just decorate its tyrosine residue with more and more glucose units and can do that up to 8 times. So, and yeah, this requires no energy, it happens spontaneously. It's pretty incredible. So after glycogenin seeds itself with this glucose, then glycogen synthase has something it can recognize. It'll come in, bind, and start polymerizing those non-reducing ends. So after we have a long linear chain, we need to branch it, and that happens with branching enzyme. Similar to the debranching enzyme, this is one enzyme that has 2 jobs. The first, it cleaves an alpha-1,4 linkage to make a 7-unit chain, and then it drops this 7-unit chain onto another glycosyl unit and forms an alpha-1,6 bond between them, and that is how branches are made. I will also mention that this is an energetically favorable reaction. It costs about twice as much energy to build an alpha-1,4 bond than it does to build an alpha-1,6 bond. So this reaction is energetically favorable and happens spontaneously. In the end of glycogen synthesis, we have some molecule that resembles this cartoon version with the glycogenin G at the center and its little 8-unit baby glycogen starter molecule there. And from there, the glycogen just kind of branches according to some very specific rules. So there's 2 branches per chain and about 13 units per branch. And this just kind of continues in an almost very fractal way till this whole molecule is created. In all, there's about 120,000 individual glucose units in a single glycogen molecule, which is a lot. That's like a huge capacity for energy storage. And also, like we mentioned earlier, there's tons of non-reducing ends. So again, all of building up this, so it has this branched architecture with all of the non-reducing ends means glycogen can be broken down rapidly to produce glucose, and then the length and number of the chains means that there's a lot of energy there, so we can sustain response. So whether you're a rabbit or you're Russell Wilson, you can recognize a threat, get on your feet, run, and keep running until the threat is gone. And again, plants have an inferior form of glucose storage in the form of starch, so they can't actually walk. The sum up of glycogen metabolism, I told you about all of these enzymes. I want you to appreciate that the degradation pathway and the synthesis pathway are overall kind of similar, and they even share an enzyme or two, but there are some very important differences that differentiate the two pathways, and you guys are going to see kind of another example of this when you learn about gluconeogenesis on Monday. It's like kind of the opposite of glycolysis, but very different in important ways. So this is, and it provides key points of regulation also. I will point out as one last thing that UTP is consumed in this reaction when you're adding glucose 6-phosphate onto a new glycogen. And we have to regenerate that UTP somehow. Thankfully, there's an enzyme that does this. The nucleoside diphosphate kinase uses ATP to rephosphorylate UDP, and the take-home message is that storing 1 glucose on glycogen costs an ATP. But that doesn't really matter, because if the body is in a phase where it's going to be storing glycogen, you have a lot of sugar and a lot of ATP around anyway, so you can afford that ATP. So that was glycogenesis. Anybody have questions up to this point? All right, well, we'll talk about, oh, did I hear a muttering? No, okay. We'll move on to talking about glycogen storage diseases now. This is that pathway. It's important. All of the members have to be functioning properly in order to meet the energy demands of the body and use glycogen to supply that energy. So, not surprisingly, a lot of enzymes in this pathway are associated with human disease. If you have mutations in either branching, debranching enzymes, glycogen phosphorylase in either the liver or muscle, and then the liver's glucose 6-phosphatase, these are all associated with disease. And in fact, glycogen storage diseases are so common, there's even an association for them for patients and parents and researchers who are afflicted by and interested in these diseases. They are a class of genetic deficiencies in any of the glycogen metabolic enzymes. So you're born with these things, and most of them are recessively inherited. They have an incidence in the US of every 1 to 20 to 25,000 births. And there are 10 different types depending on what enzyme is affected and what tissue is affected. The first thing you may notice about these diseases is that they have a common name that's named after a famous doctor or biochemist, which means that if you work really hard in this field, you too can have a horrifying disease named after you. You may also notice that you recognize some of these enzymes as being involved with glycogen breakdown, the things that we just talked about, and a few that are also involved with glycogen synthesis. We're going to talk about 4 of these diseases today. So the first one is von Gierk's disease, and this was first identified in 1929. This German doctor, Edgar von Gierk, noticed a bunch of young patients that had enlarged livers and hypoglycemia, and he named this disorder after himself, and these poor kids had this disorder. But we find out 20 years later that he had very cleverly isolated a population of patients with a glucose 6-phosphatase deficiency. This was the first example of an inherited liver enzyme deficiency that was associated with human disease, and it's the most common glycogen storage disease. So if you don't have glucose 6-phosphatase, that rids the liver of its glucose exporting abilities. It doesn't have the ability to dephosphorylate that glucose 6-phosphate. And you may remember that the muscle lacks this enzyme. So because of that, the muscle glycogen handling of these patients is not affected. It's all in the liver. But the problem is that there's massive accumulation of glycogen and glucose 6-phosphate, and this causes liver and kidney damage that causes most of the complications that these patients have are actually related to liver failure and kidney failure. They found that if you can catch this disease and diagnose it in a young child before organ damage has happened, there are some simple dietary things you can change that actually give them a pretty good prognosis. So logical things like feeding them cornstarch to maintain blood glucose, because the liver isn't going to do that for them, but cornstarch apparently really helps. And then also avoiding sugars that are metabolized directly into glucose 6-phosphate. You don't want to be making more glucose 6-phosphate if the liver already doesn't have a way to get rid of it. So sugars like galactose and fructose, they try to avoid. But again, you have to catch this disease early in order for the prognosis to be good. Anderson's disease is a branching enzyme deficiency that is also pretty terrible. Recall, normally, branching enzyme introduces branches into the growing glycogen chain. In Anderson's disease, they can still make glycogen just fine, you just can't branch it, which doesn't seem like too big of a deal, but the problem is that these very long, unbranched chains have poor solubility. So if you've ever tried to dissolve cornstarch in water at room temperature, you know that those like long, linear amylose chains don't dissolve very well. The solution stays white and milky. And a similar thing happens with these long, unbranched glycogen chains. As a result, they precipitate. Glycogen actually forms these precipitates in the liver and in the heart. Best case scenario, you get liver cirrhosis, muscle weakness, and heart problems with this disease, but it is a cause of miscarriage, and most patients who have this succumb in early childhood. Horses and some breeds of cat are also affected by an inherited deficiency in branching enzyme, so at least we're not the only ones in the animal kingdom that are traumatized by this disease. The last two diseases we will talk about are glycogen phosphorylase deficiencies. So there are two different genes for glycogen phosphorylase, one for the muscle and one for the liver, and you can have mutations in either and have two different diseases. So if you have that mutation in muscle, you don't have glycogen phosphorylase and can't break down this very first step of glycogen. So you would predict that that would cause glycogen to accumulate in muscle, and we can see evidence of that very clearly. This is human muscle samples, slices that it's a healthy one and a patient with McArdle's disease, and they've stained it to visualize the glycogen. And like, I'm not a histologist, but you can see pretty well that the McArdle's disease one is darker, and that is the glycogen that's accumulating right there. So how this translates to symptoms for a patient are exercise problems. Normally, our muscle contains a very low concentration of ADP at rest, but as we exercise, the ADP concentration increases because you're consuming ATP and also making ATP, but there's a slight increase in the ADP concentration. People with McArdle's disease have a huge spike in ADP concentration when they start to exercise, and that's probably just because the muscle is lacking this very important energy source. It can't break its glycogen down, right? It doesn't have its glucose. And this causes cramping and muscle pain and sounds very awful, but they're actually finding that after the patients are acclimated to light exercise, it's almost like the muscles find another way to get the fuel that they need, and then they can perform a lot better. So this is a really interesting corner of research that's happening in Europe right now, looking at exercise training regimens they can give to these patients to, let them be able to exercise. And then they're also finding, not surprisingly, sucrose supplementation helps these patients as well. But that's just, you're feeding the muscles an exterior form of sugar. The other glycogen phosphorylase deficiency in liver affects the same enzyme, different gene again, and it is named after this Belgian physiologist, Dr. Haers, who characterized this disease in 1959. The liver can't break down glycogen in this disease, and this would seem like a huge deal. But oddly, for this disorder, a lot of people are asymptomatic. There could be people in this room that have this disorder and you don't even know it. It can cause mild liver enlargement, mild hypoglycemia, and low muscle tone, but they find that most of these symptoms stabilize by adulthood, so they're fine, which also makes it a little bit more remarkable that Dr. Haers was even able to identify this population of patients in the first place if they're largely asymptomatic. But this kind of broke my brain a little bit until I thought about how the body manages its fuel supplies. So given a choice, any cell would probably prefer to use glucose over fat, but if you don't have enough glucose, if starvation is going on, or you can't break down glycogen in your liver to release blood glucose, the body is pretty efficient at switching over to using fat as a fuel instead. And you guys will learn a lot more about this next quarter, but one telltale sign that the body is burning fat is that ketone bodies appear. That's just a byproduct of breaking down the fat. And sure enough, these patients have elevated ketone bodies. So it seems like the body is amazingly, despite lacking this major enzyme, been able to shift its focus of using glucose for fuel to using fat for fuel. It's remarkable, yeah. So to contrast these two phosphorylase deficiencies, just so you have them clear in your head, there's two genes that can be affected differently. The muscle version causes McArdle's disease, and that causes problems with exercise and cramping. The liver version, if that is deficient, causes HERS disease, which has pretty mild symptoms and pretty good prognosis. So there are the kind of, to summarize all of those diseases, here's the pathway again. We know that all of these players have to be working together. If you don't have glucose 6-phosphatase, that results in von Geerk's disease. Deficiencies in branching enzyme cause Anderson's disease. And then we talked about 2 glycogen phosphorylase deficiencies, McArdle's that affects muscle and HERS that affects liver. And yeah, with that, I will wrap up. This is kind of the overview. Hopefully all of these words mean something to you guys by the end of today, and you can use this to study. And thanks for listening. Yay.